Welcome to Genomics Unlocked, the #1 platform for all things genomics. Today's episode focuses on revolutionizing diagnostics, whole exome sequencing with DNBSEQ, featuring Dr. Ashraf Dallol and Dr. Akbar Ali Khan Pathan.
I welcome you all and thank you all for taking the time and joining us today. Upcoming webinars that we're going to be having on the 20th of June will be Harnessing the Power of Genomics with Dr. Shang Tong and Robert Henry, and this will be happening on the 20th of June. And then on the 5th of July, we'll be having comprehensive molecular tumor analysis by Alarix, powered by MGI, with Dr. Marie-Laure Yaspo and Priyanka Karupati, who will be presenting on the 5th of July. So please join us for these upcoming webinars. We will be sending you reminders of this as well. So for today, we are talking about revolutionizing diagnostics with whole exome sequencing on our DNBSEQ platform. And I welcome Dr. Akbar and Dr. Ashraf today, and thank you for taking the time to present to us today. We will begin with Dr. Akbar, who is the Senior Field Applications Specialist Manager based in the Middle East and handles the Middle East and Africa as well as the India region. Akbar has more than 15 years of experience in the field of genomics, currently as a Senior FAS Manager at MGI. Dr. Akbar has extensive knowledge and experience with a PhD in comparative genomics, and has more than 50 research articles published in reputed journals around the world. Dr. Akbar also has quite a few book chapters, one of which recently was published, and you could look them up and get a little more information about the kind of research that Dr. Akbar is involved in. So I'll hand over to Dr. Akbar, who will be presenting today the overview of the whole exome sequencing workflow in molecular pathology, which is offered by MGI, focusing mainly on library prep methods and data analysis across our DNBSEQ platform. So Dr. Akbar, I will be handing over to you.
Rishma, thank you for the introduction. I hope you can see my screen.
Yes, we can.
So let me directly enter into the presentation. So, okay. So just to brief you about what exactly is the exome. We all know that the human genome as such is made up of 3 billion base pairs, meaning that ATGC contributes to roughly like 3 billion. And not all of these 3 billion letters provide valuable information which forms the proteins. So only 1 to 2% of the entire DNA codes for the protein, and that what we call as the coding region, or we call it as a exome. And in this 1 to 2%, most of the genetic changes are associated, like almost 85% of the genetic changes are associated with a disease, and that can be found in this exome. So in a clinical setting, like whole exome sequencing is commonly done. The better way of doing it is also to have a look at the whole genome sequencing, but as it is costly and then the coverage is less, generally we go for a whole exome sequencing in almost all the clinical settings. So this is a brief introduction about the exome. Now, moving down towards the testing technologies, what has been adopted across the world. So we do have something called the targeted testing. So targeted testing basically deals with single genes, like the one I have given an example as HPV gene. So it's a kind of single gene which roughly spans around a kb to 0.5 to 2 MB. So the regions of these genes can be sequenced using a Sanger sequencing method, and then we find out the variants across these genes. The other way of testing it is going a little bit further towards the panels, where we have 10 to hundreds of genes which corresponds to panels. And most of these panels are run on next generation sequencing machines. And these panels, for example, we have BRCA, we have myeloid panels, we have solid tumors, and we also have many other panels which have a combination of 10 to hundreds of these genes. And most of these panels, if you see, they correspond to a panel size of 2 MB to 10 MB, so what we call as the panels. And anything which is not picked up by these targeted genes or the panels are the ones which will be looked for in the whole exome and the whole genome sequencing. So when single gene or panel testing has not provided a diagnosis, basically we move towards the larger regions where we cover the exomes as well as the whole genomes. So when we talk about the whole exomes, we would like to know what will be the panel size. So most of the companies which provide some kind of exome testing panels fall in the range between 30 MB to 70 MB. So most of those panels will be in this range. And anything beyond this will be the whole genome sequencing, where we go up to 3 GB of the data. Now, when we talk about the whole exome sequencing methods, so what are the different methods we adopt to do the whole exome sequencing? So the first target enrichment strategy was on a array. So basically, these single strands of DNA were used as probes onto a solid matrix, and onto which were actually about this targets, and then they were captured based on the hybridization methods and later sequenced on an NGS method. So over a period of time, as these methods were expensive, involves a lot of hardware and also needs a larger DNA input, they were replaced by something called the solution-based capture method, which has gained popularity in the recent years. So basically, if you look at this right side image where we have the complex biological samples, we extract the nucleic acids, we do some kind of fragmentation using either enzymatic methods or some kind of mechanical shearing. We make the libraries using the DNA or the RNA probes, and then we hybridize them onto our targets, and later on we sequence them on the next generation. So this is overall the workflow. Now, what depends on a stable hybridization is the homology, which is between the target, that is our nucleic acid, and the probes, what we call as the DNA and RNA bait here towards the right side. So the degree of homology between the target and the probe results in a very stable hybridization. So that's what we call as a stable hybridization. And most of the limitations for the exome sequencing relies on how effectively we capture this particular regions. So this is a basic introduction about the methods. Now, when you ask me which probes are better, whether the DNA probes or the RNA probes, well, it depends on many things like the sample type, the application, the target region coverage, the capture region efficiency, and so on. So to best explain, I think RNA baits cover greater portions of the exome. So if you are interested in not only the exonic regions, but also some kind of intron-exon boundaries, as well as some intronic regions, we always prefer a RNA probe. And if it is mainly focused on the regions of genome, smaller regions of genome, we always go with the DNA probes or the DNA bites. A very good example of what sort of a probes to be used is well explained in this publication, where they have compared 4 types of target enrichment baits for exome, and you can have a look at this publication to have a better understanding of which probes suit better for your research purpose. Moving on further, what exactly a comprehensive exome panel looks like. So here, the comprehensive exome panels target roughly around 36.8 MB of the human coding regions, that's the exon, and which covers almost 99% of the publicly available databases like ReferSeq, CCDS, GeneCodes, etc. And apart from this, most of the companies also target on the common features, which are the bait type, where it is a RNA-based probe or a DNA-based probe. What should be the ideal length of this probe? And then what will be the length, the total length of this probe, and what sort of a target length it captures when it is put on the target, what sort of fragmentation method they use, whether it is enzymatic or mechanical, and what should be the concentration of the input library in terms of nanograms, and what databases, how much of the databases does these panels cover. So these are the common features that most of the companies will look into And we have a wide range of exome capture panels and kits available on the market, basically based on all these common features. Now, when we compare the exomes, we can divide the exomes into two categories, one which is purely based for clinical purpose, and the other one which is for the research use. So the blue highlighted part here represents the clinical exomes, which basically target the clinically relevant genes. And what should be the size? They have a panel size roughly between 4,000 to 8,000 genes with a panel size of 8 to 12 MB. We also have something called the flash exomes, which go a bit less than 8,000 genes and a smaller panel size. And if you go larger, capturing a larger gene size, then we have something called the comprehensive clinical exomes, which target roughly between 10 to 20,000 genes. And the panel size will range from 20 to 40 MB. Okay. Most of the publicly available reference databases where we have well annotated variants are actually covered under these panels, which are ReproSeq, GeneCode, CCDS, HGMD, CleanVar, etc. And most of the molecular diagnostic companies are the clientele here. And if you come to the green part, which is the research exomes, not only those traditional genes or the traditional panels which are mentioned above are included, but they also include other regions which target various diseases involved in reproductive health, neonatal care, cardiovascular, hereditary tumors, monogenetic disorders, some kind of personal genomics, pharmacogenetics, and then also do with the mitochondrial defects. So when we talk about mitochondrial, obviously the panels what we have from MGI do include the mitochondrial region with 100% coverage. And some of the commercial companies also provide these mitochondrial panels as spike-in panels, where you can add this spike into the existing exome capture methods. So this is something to do with the clinical versus the research exomes. Now, coming to a population like what we have in Middle East, where consanguinity plays a major role, I think we need to have a combination of both clinical as well as research to come to a conclusion on certain disease-causing variants. So moving forward, like what are the solutions what MGI provides are basically explained in this slide. So we do have two panels, which we call as the V4 and the V5, which are basically research exomes. And the main difference between these two panels is the V4 is a bit smaller panel when compared to the V5, which covers around 59 MB of the capture region, whereas the V5 targets around 69 MB. Both are again RNA-based probes are being used here, and most of the databases they cover are pretty much the ones which are covered by the other clinical panels. And with the V5, along with the well-annotated reference databases, we also have our own MGI's own database, which covers at least like 53.36% of the database. So this is something which is an added advantage of having a MGI panel. And how many genes are covered? For the V4, we roughly cover 20,000 genes. And for the V5, we have 25,000 genes plus 100% of the mitochondrial region. As you know, mitochondrial region, I think in the present scenario, there are a lot of publications coming up with the significance of mitochondrial genome, basically in the neurodegenerative diseases and most of the cancers, basically because of the multiple copy number they carry. So this is something which is good for mitochondrial regions as well. And how many number of reactions can be done in a single, how many number of samples can be included into a single reaction? Both of the panel can take up to 8 samples into each of the panel. And the V4 takes roughly like 10 GB of data per sample with a 100x depth, and 15 GB for a V5 panel with a 100x. And if we look at the on-target basis, we have got roughly like 95 to 96% of the target basis with a 20x coverage. And the capture efficiency falls somewhere between 60 to 70% for both of these panels, with a duplication rate of roughly 10% and lower. And along with this, we can also have our own analysis servers, which can convert the FASTQ files to VCF. And we can also put the third-party softwares which can generate the valuable clinical report onto our servers. Moving forward, like a wide range of databases are covered under our panel. And on the left side, if you see, there is an example of almost like 450 genes which are covered with the MGI V5 panel. These are the fully covered genes. And it's very clear that from the ref gene database, almost 450 genes have full coverage. At the same time, if you look towards the CCDS database, we cover 2,116 unique genes which are under CCDS. And just taking one single gene as an example, we have here something called the BBS10 gene, which is Bardet-Dial syndrome. And if you see this, this is basically a protein complex involved in chaperonins for protein folding. And a defect in this particular gene will cause some kind of retina damage, kidney and liver damage, as well as the brain. Okay. So if you look at the ClinVar database here, so these are the actionable variants which are mentioned. And in the V5 panel, it's covered with 100% accuracy. So this is just one example. And this particular BBS10 is widely known for its prevalence in consanguineous populations, like the one which we have in the Middle East. And apart from those mitochondrial regions, as well as the full gene coverage, we also include in our V5 panel many other smaller panels, what we call as the sub-panels. So you can see here, we have got different panels included under V5. So we have something for diabetes, neonatal care, cardiovascular, congenital heart disease. We also have for thalassemia, which is covered, mitochondrial disorders, autism, pre-implantation genetics, neuromuscular, as well as monogenetic disorders. So along with this, in the panel, like majority of these sub-panels are actually included. And if you want to know what exactly are the regions which are covered under these panels, you can go into the link which is provided below, where you will find the bed file for most of these regions which are covered. And you can zoom in and see which regions are covered, and if the regions do add some kind of a diagnostic value for your regions or for your testing. Moving on further, here is a comparison between MGI V5 panel versus the other vendors. So highlighted here, the first one, the dark blue, is the MGI V5 panel. And if you see here, it has covered almost all of the base pairs which are mentioned in the CCDS database. And accordingly, you can also see that they are equally present in the gene codes, MIR base, and then ref gene, as well as UCSC. And towards the right, if you see, we have got 100% coverage of our mitochondrial genome by the V5 panel. And apart from this, we also have coverage in ClinVar and HGMD. So if you see here, most of our panels do cover 95 to 96% of ClinVar, as well as the HGMD database. Coming to the performance of the V5 panel, if you see here, this is the coverage plot onto your left side. And you can see that more than 96% of the region has greater than 20x mean depth coverage with the MGI V5 probes when compared to the other vendors, vendor 1 and 2. Towards the right, you can see the SNP. Towards the right, you can see the SNP precision and the SNP sensitivity. So the first blue line here, the dark blue, is the V4, and the subsequent one is the V5. And you can see the SNP precision ranging from 99% and above for the SNP precision. And for the sensitivity, the V5 is much better than the V4. And if you see the indels precision and sensitivity, obviously because of the smaller size, the V4 has got a good indel precision, again greater than 90%, less than 94 when compared to the V5. Same is the case with the indel sensitivity, which is towards the right. So the V4 has got a better indel sensitivity when compared to the V5. And the simple reason is like the smaller the panel size is, the indel sensitivity and precision actually will be higher. Coming to the duplication rate and the capture rate. So if you look at the duplication rate, the V5 has a very good duplication rate, which is very less, like 3.73%. And then if you look at the capture rate, we always have a capture rate which is between 60 to 70%. So this is very good. And then if you see the coverage, we start from 0 to 20x, you see that most of the V5 on target coverage is roughly above 96%. And when we talk about the how many samples can be multiplexed onto our NGS sequencing platforms, I've given you a rough calculations towards the right side on the table, and towards the left, how do we calculate how much of data is required per sample? So we do have a small formula here where we take the panel size, and for us the panel size, as you know, for the V5 it's around 69 MB, and we take the depth and then we divide it by 1000. 1000 is nothing but converting the GB to MB. And then we have got the capture rate, like we have discussed, our capture rate is somewhere between 60 to 70%. So you do the capture rate calculation here, and then you minus the duplication rate from 1, and then you get how much GB of data is required per sample. So in this case, if you see here with a 100x coverage and PE 100, with a 60% capture rate and a duplication rate of 10%, we need 12.7 GB data. And in the same way, with a capture rate of 50%, you can have a per sample data of 15.3 GB. And if you see here for a G400, we have got like each lane like 450 million reads. We calculate how many samples can we put onto a single lane. And if you see here, we can put up to 28 samples onto a single lane into we can put up to 28 samples on 4 different lanes, assuming 7 samples per lane. So this is how we do a basic calculation, and this includes a 100x coverage. And if you want to go beyond this, we do have a range of sequencers which will provide the required output. So with the G99, which is a small benchtop sequencer, we can go up to 2 samples, generating at least 20 GB data per sample for a 150x coverage. In the same way, on a G400, you can run up to 27 samples per flow cell into 2 flow cells, so roughly 54 samples. And on a T7, as you know, we have got 4 flow cells, so each flow cell can accommodate up to 70 to 75 samples, depending on how much coverage you want. And roughly in the 4 flow cells, we can generate somewhere between 280 to 300 samples in one run. So this is the throughput for our sequencers. Now, once we have the data generated, so what exactly will be the data analysis steps? So what our systems or the sequencers do is they generate some kind of a raw image where we load our DNBs, and those DNBs are actually captured. The images are registered in the second step, and moving forward, the intensities for all these bases like A, T, C, and G are actually calculated for each DNB. And then once these intensities are calculated, they are corrected. Say, for example, if you see here at the bottom, the raw intensity, we do have signals coming for A, C, G, and T. This has to be corrected to identify the right base. So what we do is we do something called the intensity correction. And once the intensity correction is done, the software will be able to call out the right base. In this case, it's a G. And then this will be recorded into a FASTQ file and then further reported. So the system basically has the capability to generate the FASTQ file. So on a G400, we have something called the base called light, which generates the FASTQ files. Whereas on a T7, along with this T7 instrument, we also provide you something called the Zetron Lite, which is an additional server which generates the FASTQ files. So if you want to have more number of samples to be processed in a lesser time, we provide something called the Zetron Pro, which does the analysis in a much faster time. I will talk about this analysis time in the next slide. So this is what basically the servers do. And apart from this, the secondary files can be generated right from the FASTQ to the VCF. We provide you Zetron Pro, which has got source tools like Minimap2, BW8, GATK, etc. And these are the ones which generates the VCF files. So the external VCF files are generated on the secondary analysis pipeline, that is the servers. And onto these servers, we can also have our third-party softwares which generates the clinical reports. Like most of the cases, we have got third-party softwares like Seq1, Seqmax. So all these softwares can be installed on these servers, and then it generates the clinical reports. So that's under the tertiary analysis. So basically, the system generates the cal files, and the Zetron Lite does the FASTQ writing. The FASTQ files enter into our Zetron Pro, and then they get converted to a VCF file. And on the VCF file, you can have all your third-party softwares to work on, and then it generates a clinical. So this is the overall picture. And then if you look at the performance of these servers, so if you have a Megabolt, which is basically a server which is provided with the G400 to convert the FASTQ files to VCF, you can see that it roughly takes 1.96 hours if it is a WGS with 30x coverage, and if it is WES with a 400x coverage for germline samples, it roughly takes 41 minutes. And if you use the same file on a Zetron Pro, it goes for as low as 4 minutes to process one sample. Same is the case with somatic when we have a 400x coverage. And then if you see here, it roughly takes 8 minutes for a Zetron Pro to do the analysis, and the Megabolt takes roughly an hour for sample. So most of the samples which are processed on a T7, you can have this analysis done in less than a day. So apart from this, like that was the discussion for the V4 and the V5 panels. Along with these panels, we also have tested most of the commercially available clinical exomes coming from different third-party companies. And we basically do this with a single step called conversion. So most of these double standard libraries will be actually circularized so that our sequences can read the circular libraries, and then we circularize them with using our app conversion kits. And once we circularize them, we sequence it onto our sequences. So basically, the table here explains about those particular third-party clinical panels which have been extensively validated, and we have generated good app nodes on most of these clinical solutions. So if you look at here, you have got the 100x depth, and each sample requires how much data, and what sort of on-target coverage it is, and then what will be the capture efficiency, how much of the duplication rates are covered, and then we also have some examples of the third-party interpretation softwares which can do the clinical reporting part. Now coming to the V4, V5 product information. So like if you see here, it basically depends on the number of samples you wanted to put onto our sequences. So if you have some sort of more number of samples to be included in a single run, we always suggest you to go with our dual barcode here. So again, this depends on the application and the throughput, what the lab requires. So if you see here, most of this NGI Exome universal library prep sets go with a single barcode, and whereas this universal library prep sets with UDB goes with the dual barcode. So we have got two sets of adapters here based on the number of samples you wanted to sequence. And if you go down further, and if you have some kind of a clinical samples where you need to use FFPE, we also have a kit for that, which is the last one here, MGI Easy Duplex UMI Universal Library Prep Set. So this can go up to like 10 nanograms of DNA. So most of the FFPE samples which fall into this range can be sequenced using this kit set. Coming to the ordering information, like I have told you, we have got two probe sets, V4 and the V5, and this is the part number. And you have got this capture kit, which will be provided. The universal library prep set can be used for sequencing your libraries on different sequencing platforms like Illumina and Antorrent. But if you are more specific and you have an in-house MGI platform, you can always use our own FS library sets, which are compatible with MGI. The beautiful part about the MGI's technology is we also provide you a complete automation where the libraries can be made on our automation systems like the SP100 and the SP960, which are cost effective. So these things can be included as a part of the workflow. So in conclusion, I would like to say this, that MGI provides a total solution for whole exome sequencing, right from the sample extraction, and then the library preparation and the DNB generation. And we also have a range of sequencers which can provide you different throughputs. We have got the servers which can do the primary and the secondary analysis. And finally, all of this workflow can be integrated into the existing hospital LIMS system through our ZLIMS. So that's one part which can also be integrated. Apart from this, I think we also have some recent publications in Nature's scientific reports. So two of them I have just added here as an example. So one of this on the left is the recent publication which we have published from University of Jammu, and you can see that they have discovered a novel pathogenic variant in a disease called neurofibromatosis. So this is basically a familial study where they have screened 3 generations of families in Jammu and Kashmir with a multiple affected family members showing clinical indications of NF1. And I think this is something to hint you that the V5 can be exhaustively used for discovering pathogenic variants. And towards the right, we have another example of a paper where MGI sequences have been used, and the publication clearly says that the reliability and quality of the data which were obtained by our sequences was unmatched. So these are the 2 publications which we have, and I think that's pretty much from my side. Thank you for your time, and if you have any questions, I think I can answer you at the end of this session. Thank you.
Thank you, Dr. Akbar, for that informative presentation and introducing whole exome sequencing on our platforms. We have one question here from Eugene. What is the effective capture rate of the MGI V5 probe with your library prep protocol?
Yeah, it's 60 to 70% what we say, but again, it depends on the quality of your sample. 60 to 70 is what we have.
Okay.
So I have yeah. So you can see the capture efficiency, it's up to 70%.
Great. If you have any other questions, please feel free to. Okay, we have one more. What is the minimum NA requirements for this whole exome sequencing? This question is from Sudhir.
Minimum requirement in terms of sample?
Yes.
So if you see here, so this is actually the samples minimum input requirement. And again, this depends on the type of kit which you are using for the library preparation. Like as I have told you that we do have a range of kits which take different input concentrations. Like if you see the UTB here, it goes from 1 to 1000. And there is one more which goes from 10 to 100. So 10 to 1000. So depends on what type of sample you have, and then how much concentration of DNA you have got, and what is the quality of that particular DNA sample. So based on that, you can decide which library kit to use and what sort of adapters to use.
Okay, and so this was asking, what about cell-free DNA?
Again, like cell-free DNA, if you really want to go, I think you already have smaller panels like we use for the low-pass whole genome sequencing. So that could be one option. And still, if your concentrations falls in this range of input amounts required, we can go with one of these groups.
Okay, and then we have another question from regarding which are better, RNA baits or DNA baits?
Great question. So again, for a discovery panel, I think if you see, that was the discussion which we started with. So if you look at here, most of the RNA probes cover greater portions of the exome. Means if you have regions which are like, apart from the exons, you also wanted to sequence some kind of intronic regions which have clinical significance, then definitely RNA probes will be the ideal ones. But remember, when you go with the longer probe size, you also will lose some kind of sensitivity. So if you compare this part here, the longer probes we go and the longer regions we cover, I think here, sorry. Yeah. So if you see here towards the right side, so the indel precision, as you have a longer panel, it actually decreases. So from the V5 to V4 to V5, the indel precision is dropping. So basically, this depends on the resolution which you wanted to have on a longer panel. So it depends, like if your regions fall pretty much in the conserved region and DNA probes can do the job, it's always good to go with the DNA probes. And if you think that it has to cover some kind of intronic and exonic boundaries, it's always good to go with the RNA probes.
Okay, and when you say capture efficiency, do you mean target rate?
Yeah.
Okay. I think those are the questions that we have so far. I encourage the audience at any point during the webinar, if you have any questions, please put them in the question panel and we can answer them at the end of the presentation. And if you need any more information about any of our products, whether it's the sequencing platforms or the kits, a lot of the information is available on our websites. And you can also reach out to us at any time and we'd be happy to help you with more information. So we don't have any more questions, so we can move on now to Dr. Ashraf. Thank you, Akbar, for the presentation and for answering the questions.
Thank you.
We'll be moving on to Dr. Ashraf. I'm just going to quickly give Dr. Ashraf an introduction and then we can begin. I take this opportunity to welcome Dr. Ashraf from NoorDx, based in Saudi, for joining us today and taking the time to talk about Dr. Ashraf's experience using DNBSEQ technology and working with MGI. Dr. Ashraf has a PhD in biomedical sciences from University of and has been working as and worked as a postdoctoral position in Birmingham after that. Dr. Ashraf then joined King Abdulaziz University in 2008 and was there until 2021 before joining NoorDx now as the Diagnostics and Discovery NoorDx Diagnostics and Discovery Laboratories as the lab director and chief scientific officer. So Dr. Ashraf has 20 years of laboratory experience in using genetics and epigenetics technologies and approaches, and has been part of more than 60 scientific publications across a range of journals around the world. And now Dr. Ashraf works with NoorDx and NoorDx customers to harness the power of emerging technologies like that of DNBSEQ to further enhance molecular biology and its applications for personalized medicine in the Middle East. So today, Dr. Ashraf's presentation will be providing an overview of NoorDx's experience with providing a clinical whole exome service in Saudi Arabia, utilizing the MGI platform, setting an accredited diagnostic service based on whole exome sequencing requires a multi-step approach in order to facilitate delivery of accurate results on time. And I hand over to Dr. Ashraf now to take the floor and talk about NoorDx's experience.
Yes. Hi, good afternoon, everybody. Sorry for that, Reshma. Thank you for the introduction and for the invitation to speak to the audience in this webinar series. This presentation is going to be slightly different from what Akbar just mentioned, and he actually took a lot of the burden from my presentation and took comprehensively about the technical details involved in exome sequencing. So I'm going to focus today about showing or presenting our experience at NoorDx in utilizing whole exome sequencing and offering it as a clinical service, as a commercial clinical service, which we are the only lab in Saudi Arabia, the commercial lab in Saudi Arabia, that offers this service end-to-end. So, not further ado, see. So our company started officially in 2008, but with the COVID and the crisis, it was delayed, and it started started with COVID testing, and we used in-house from cast reagents and master mixes, and managed to offer the COVID test to over 100,000 cases in 3 months. At like 10% of the market price, which helped the community to test more and prevent the disease spreading even more. And across the year, we were working really hard on establishing the platform. And in March 2022, we got the platforms, and in June we started the first whole exome run. And in December 22, we got the CAP accreditation. So it was a quick journey and a lot of hard work, a lot of teamwork involved to establish these KPIs. And right now we offer an end-to-end service. Sorry?
Your presentation is still in presentation mode. If you can put it in full screen so we can see the slides. Yes, that's great. Thanks.
And now we have an end-to-end service from sample collection towards reporting, all done in-house. And this is it has been a challenge, a new, relatively new approach in Saudi Arabia, because most of the genetic or genomic sequences is done abroad or in very specialized hospitals. So this has been our mission from the beginning, and we believe that we reached a good stage at that. And for the accreditation, we have worked to get everything available, starting from Ministry of Health, CAP, Waqaya, and Sibahi, and soon it will be the ISOs. So it's important to as a startup company, to show the customers that you are offering reliable service, a credible service, as judged by your peers. Okay, the question of this talk is how to establish a diagnostic West service. So I think a lot of us agree that to establish the service, you need several components, include that, namely the team. I believe you have to have the team before you do anything else. Then platform, bioinformatics solution, the validation approach that you need to set up to make or streamline your approach and make it credible as a diagnostic service. And then to show that you are credible, you go for the accreditation. That goes two ways. That has the benefit of catching mistakes before you offer the service, and correcting them quickly and efficiently before producing diagnostic reports. So in terms of the team, we have our core mission was capacity building of Saudi technical staff, and at NoorDx, 90% of our staff are Saudis. They are graduates of local universities, and they have been trained on the variety of techniques at NoorDx, from sample preparation to NGS development. And this, we are proud of that, that we have that kind of localization of the service achieved at a very short time. And also, in addition to the technical team, we have board certified genomic scientists, including head by Naif al-Muntashiri from Taiba University. We have Sharif Hala, who's overseeing the metagenomics side, as well as others. Now, for the platform, when you start up a company to offer a diagnostic service, it's a difficult choice, which one to go for. I mean, this is a slide showing the options available. And now I can add 2 more lines of options. We can add the element bioscience machines. We can add even PacBio offering short-read sequencing. And on top of that, what do? You rely on short-read sequencing or long-read sequencing. So which one to go for? If you have a lot of money and you don't worry about the operation cost, you probably go for the big beast like the NovaSeq or the T7. But for a startup and the flexibility, you need the flexibility when you start up a diagnostic service. You need to offer amplicon sequencing as well as whole genome sequencing, and offer that at a cost that will not break you as a startup. And for that, we opted to achieve that. We found out that the DNBSEQ G400 is probably, in my opinion, the most flexible platform out there, because from 16S amplicon sequencing to whole genome sequencing, it does that. You can do 1 lane, you can do 4 lanes, you can do 8 lanes. And the turn-on costs are competitive, and you don't have to break the bank to operate the machine. So in terms of a commercial, in terms of diagnostic, in terms of running as many validation studies as you can, we found out that this platform is the best out there because it gives you the flexibility of doing whatever you want. Right now we've done RNA-seq, metatranscriptomics, amplicon sequencing, whole genome sequencing on the same machine, and we always found that the data quality and size, and trust me, I'm not paid by MGI or anything. This is from real life experience, and I have to be to say this machine is probably the best platform to start with. And as Akbar showed, the accuracy and streamlined data analysis is really good. And in Saudi Arabia, the technical support has been magnificent. It's a machine that's not difficult to repair if it breaks down, and this altogether, such factors makes it an ideal platform to start with. Then, if you have the team, the platform, now what to do with the data? How do you analyze it? Especially, how do you analyze it in a diagnostic setting? The difference between analyzing data and using bioinformatics for research and diagnostics is you both use the same tools, namely, but for research, you have flexibility. You don't really worry too much about reproducibility of your pipeline in the research setting, because the bioinformatician in the research setting will apply pipeline version 1 for the first research project, then a couple of months later, then he or she will have a version 1.01 of that pipeline. And for research, does not matter. For diagnostics, it does matter. Every time I change a command in the pipeline or I change a script in that pipeline, I have to run a validation series. So this was key in deciding which bioinformatics solution to use in diagnostic setting. Akbar mentioned the Megabolt solution. The Megabolt hardware is magnificent, but the software itself, it utilizes the free tools, Gattaca, BWA, so which everybody with bioinformatics knowledge can run. But again, the research setting versus diagnostic setting, that drove us to select a third-party software, and we found out the best combination was CLC Genomics Workbench, because it allows you to run whole genome sequencing on a run-of-the-mill workstation. It does not need specialized hardware, as well as QIAGEN Clinical Insight for variant interpretation and database searching. So the combination of these two allowed us to stabilize or benchmark our bioinformatic analysis of the OS exome sequencing, and that ensured that when we do the validation, we don't really need to worry too much about the changes in the bioinformatic pipeline, and it is stable, it's reproducible between the runs, between the between the samples, and so it gives us the assurance of the quality of the data. Then after you have the team, the platform, you have the solution, the bioinformatics solution, you run the validation series, the clinical validation series. Akbar in the previous presentation mentioned the differences and how the accuracy, sensitivity, specificity of the V5, V4, which is great amongst the other in comparison to the competitors. However, when you go for a CAP accreditation, you need to run a slightly different validation approach. And it has to cover several aspects, which I'm going to talk about next. But before that, why do we focus on WES? Akbar covered really this kind of analysis, so I'm going to go through it very quickly. Why WES, not whole genome? It's the cost effectiveness. That's it, really. It's the cost effectiveness. It's the ability to offer an affordable solution to the masses. And but it has a problem, which is the hybrid capture methods. Always, anybody worked with them, they know that whatever the panel, whatever the kit, they have, it has some drawbacks, but when you increase the coverage, it can override that. Then whole exome sequencing, it's the cost-effective approach because you focus on potential disease coding genes. As Akbar mentioned, it's 2% of the whole genome, so you can afford to run the sequence at 100x, 200x depth, and reliably find most of the mutations involved. And what are the clinical indications for genomic testing? I'm going to go through this very quickly. We have undiagnosed genetic conditions with broad clinical heterogeneity. So panels may be out of the questions because you don't know where to start. So you have your whole exome sequencing to cover as many undiagnosed genetic conditions as possible because you cover as many genes as possible. And this is where the number of genes is important. You could go for the flash 1,000 genes or 2,000 or 8,000 genes even, and gives you a very good diagnostic rate. However, because the broad clinical heterogeneity, you will miss some variants or in genes that are yet to be 100% clinically indicated, candidate genes, for example. Then you have neurodevelopmental disorders. WES is very good in offering diagnostic solutions for it, including intellectual disability, autism, and epilepsy. Pediatric disorders, if you combine newborn screening programs where you offer the biochemical approach, which is in Saudi Arabia at least, it's limited to 24 diseases. Instead, if you do that, we're using WES or whole genome sequencing, you potentially can cover the vast majority of those medical conditions affecting the newborn. So this is, I'm sure, if you're aware, there's a debate going on with whether we should measure every or do the exome or the genome of every newborn. And this is going to be the norm, is doing the genome of every newborn instead of biochemical analysis. And additionally, personalized medicine, basically pharmacogenetics, utilizing the WES information for pharmacogenetics or for identifying factors that influence drug metabolism and drug response, as well as, and this is becoming increasingly used in Saudi Arabia, is family-based genetic studies, where you offer an on-demand carrier testing or premarital test to identify potentially disease-causing variants. It has its own problems because you only limited to reporting the pathogenic and likely pathogenic variants. But what about VUSs? This is a big debate about reporting the variants of uncertain significance in asymptomatic individuals. However, it's at least you can reduce the impact of those pathogenic or likely pathogenic variants. And just a thought to give a taste of what WES can be useful for. For example, this is case one. quite an old case. Case of developmental delay, epilepsy, and hypotonia in a 1-year-old male. The family history was a bit lacking, but there was hardly no family history. However, there was consanguinity. We applied whole exome sequencing in on that family and identified HSD17B4 as the gene with the mutation, Endel mutation that was confirmed by Sanger. And based on that, we have a diagnosis now for that particular case, the D bifunctional protein deficiency. And that you can see the heterozygosity in the parents and the homozygosity in the child in this autosomal recessive disease. Another case quickly, here we use trio analysis and for 2 sons with microcephaly, global developmental delay and delayed speech, dysmorphic features, myopia, and signs of ADHD and autism. It's quite a big list of phenotype. A trio sequencing, trio exome sequencing, where we sequenced the parents as well as the eldest child. We identified a splice site variant in VPS13B that was homozygous, as confirmed by Sanger sequencing, and that led to the diagnosis of Cohen syndrome. So at least for these 2 cases, or cases similar to this, where you can see the phenotype is quite broad, and many genes are involved in such symptoms. Whole exome sequencing was very useful in identifying the mutation. Okay, back to the validation. Sorry, I moved a little bit from that, but back to the validation. The validation I'm presenting in the next slides utilize the methodology of using enzymatic fragmentation, using the MGI-EZ fsDNA library prep set, along with the capture probe set, the V5 capture probe set, and the DNB sequencing was on the G400 using the PE150 FCL long. We have chosen the throughput of 24 samples per flow cell. The reason is it can take more. However, we found out that the 24 number is magical because, at least in our hands, the barcodes for the MGI, it goes well in sets of 4 or 8. So the number of 24 was a good compromise. And why V5? Well, when we started whole exome sequencing, we had the option to choose any one of those. This is a list of probes used in the performance testing in CAP. And you can see SureSelect is probably, adjuvant SureSelect is the most widely used, followed by IDT. Illumina, I tried to avoid it. And then you have other probes. And then you have the MGI, V4 and V5. All these are used in the clinical setting, and participants in CAP performance testing program use. As Akbar said, the probe length is 90 nucleotides, and that has major advantages, some disadvantages. In my opinion, the difference is not really significant. But the covered region size is really important, the 69 megabase. As far as I'm aware, this is the biggest covered region. And because we are studying, or we are servicing a community where there are a lot of undiscovered genetic diseases yet, which we assume there are because of the high consanguinity. And we applied the research preferred method in a diagnostic setting. If I was commercial setting only, and just offering the diagnostic setting based on other ethnicities data, I would go for the 30 MB. That's great. It will diagnose the majority of the cases. However, when you want to study a poorly studied or poorly tested population, you go for the biggest probe set, biggest size of a probe set to include as many variants, as many genes as possible. Even if you don't use them in a reported case, you build that knowledge base. So eventually, that will lead to the discovery of candidate genes of genetic variants that are more predominant or more specific to the population in Saudi Arabia. One other question was, does it include the mitochondrial DNA? Yes, as Akbar said, it's 100%. I didn't show the comparison with the competitor here, but the coverage is uniform and significant without spiking and without really any interference. And another feature of the V5 that I personally find very important, because when previous life I was using the Illumina and SureSelect, it was always difficult to diagnose diseases like alpha thalassemia. Alpha thalassemia is caused mostly by deletions of the HPA1 or HPA2 genes or both. Okay, but when you do exome sequencing using other probes, you cannot really find, or you cannot diagnose alpha thalassemia using those probes. However, using the V5, we find out, because it covers a bigger region, we find out, yes, because the HPA1 and HPA2 reads are properly mapped, and therefore we can diagnose alpha thalassemia, because you can clearly see the deletions in this particular case, which we confirmed by MILPA. So that was pleasing to see, because it's this will increase your diagnostic rate. For a diagnostic service, this is very important. You don't have to miss out on the diagnosis of deletion-based diseases like that suffer from genes with high similarity, like HBA, SMA. We are still looking at the SMA, whether it can diagnose SMA or not, but the signs are good that SMN1, SMN2 diagnose the SMA using WES and V5. In terms of actual sequence accuracy, we use the gold standard NA12878 reference sample, or HG001, and in our hands, the precision was 97%, the sensitivity was 98%, and this was really good to see and give us the assurance of our quality of sequence. And over 150 samples, you can see the mean Q30 from the G400 was 91%. The mapping rate was 99.8. The percentage of over 20x was 99.6. Mean target coverage, because we used less number of samples per flow cell, we routinely get over 150 x coverage. And the capture rate, we reached our average was 71%. So we routinely see 70% in our capture rate of the V5. Another validation approach is to sequence known cases, diagnosed cases, or cases with known variants of different types, and clearly see the homozygous SMV sequence, an indel sequence clearly, and then the most important one, or very important as a commercial service, is to be able to offer to do CMV calling at the highest accuracy possible for an exome service. And compared to array CGH, samples that have deletions or amplifications detected by array CGH can also be detected by the V5. So that was very reassuring to see that the V5, along with the CLC, can call CMVs at a good confidence. However, every CMV requires validation, and unless you have MILPA say for every single CMV you find, we do it as a screening service at this stage. Okay, so you have the team, the platform, the bioinformatics, the validation done. Now you go for the accreditation. And as I mentioned in the beginning, we went first with the CAP accreditation because it's the most comprehensive. It requires the actual wet lab, as well as the analytical approach, as well as the paperwork and the SOPs, etc. And that was great to have at the very early stage. And then our local Saudi Central Board for Accreditation and Healthcare Institution, as well as the regulatory organizations here in Saudi Arabia. So altogether, we started our first sequence in June 2022 until December, January, we got all the accreditation. If you plan ahead and you do your homework in terms of preparing the validation studies, preparing your paperwork, you choose the best probe set and the technical pipeline as possible, the most cost effective as well, you will achieve the accreditation. You will achieve a very good service. And in the final few slides, I'll just mention over 150 cases we of WES used so far on congenital disorders in Saudi Arabia. And you can see the word clouds. The biggest words or the most frequent phenotypes we see is developmental delay, hypotonia, dysmorphic features. So I thought I'll just present the success rate of diagnosing cases with hypotonia. And we utilized the classical or typical approach of DNA extraction library preparation on the V5, sequencing, FASTA-Q, and variant calling, annotation, the QCI, and then candidate selection and Sanger validation. And doing that, we have diagnostic yield of 40% of the cases have pathogenic or likely pathogenic variants, 40% have variants of uncertain significance, and only 20% as negative. Basically, we couldn't find anything that can explain the disease. And this approach allowed us to identify known and novel genes associated with hypotonia and developmental delay, and the list is quite relatively big in terms of the number of genes, although we found one variant that is recurrent, and this is actually SK2 variant that is a founder mutation in Saudi Arabia for global developmental delay, leukodystrophy, hypotonia, etc. So that is reassuring to see that we can diagnose as many cases as possible. You can see the variants interpretation. It ranges from pathogenic, VOS, and likely pathogenic, so quite a good selection of interpretation. And the disease is diagnosed, it's all in OMIM, and ranges from autosomal recessive to autosomal dominant, X-linked recessive, etc. And at the end, I welcome any question. Apologies if I was going too quick, and I'm happy to answer any question you have. Thank you very much.
Thank you, Dr. Ashraf, for that valuable insight into the commercial side of whole exome sequencing, and especially in a CAP-accredited lab. That was very informative. We have one question from Henny at GSI Lab from Indonesia, who wants to ask about the validation using NA12878 as a control. Do you replicate control sample 150 and use it during your validation test?
I'm not sure what's control sample 150, but we use the NA12878 every time we have any change in the protocol, like an updated software or an updated kit, a change in the wet approach. So you have to use this benchmark to make sure that your change of protocol did not affect the quality of procedures, quality of analysis. So this is the typical approach we use.
Okay. So far, okay, so we have one more question from Sudhir, who says, wonderful work, Dr. Ashraf. Do you have any specific panel or test that checks for all pathogenic variants, those that have been detected through SGP?
So the Genome Project, unfortunately, the official results of the Saudi Genome Project, they're not out yet. So there aren't a commercial panel per se that you can utilize. However, from our experience, I mean, samples from the Western region, the founder mutations are rare. So it's likely that you'll find variants in every single gene or in every disease. So using a small panel may not be, in my opinion, indicated in this case. So it's better to use a bigger, more comprehensive panel for until you start seeing variants repeating in your population, in your samples. And this is, I think it will be a while before we start seeing that, at least in samples in our region. So my advice, no, there are no panel for the SGP yet, but go for whole exome at least, and that will make sure that you detect your variants. And the cost nowadays for big panels and whole exome sequencing is not big. The difference is not big in terms of cost, so you can afford to do whole exome and be more comprehensive.
Okay, and then we have another question from Dr. Sarjan Shah, who's asking how many samples are you running in a single chip, and what was your experience like with multiplexing?
A single chip, flow cell, we go up to 24 because of in our hands the barcoding distribution. We can go up to 28, but 24 has been ideal for us. Every lab has to run their initial series, start maybe with 16, and then go up gradually to see the maximum number of samples. For the PE 150 or 100, and I'm talking about. Every machine have its different output, but for the G400 P150, the 24 is ideal number for us with the V5. Of course, if you use smaller panels like Twist or Seq or whatever, you can run a lot more.
And then how was your experience with multiplexing?
Multiplexing, so far okay with the MGI probes, as long as you follow the protocol, which is make sure that you use sets of 4 and 8. You see what I mean? It's in the back end of the manual. You will see how you do the multiplexing. If you do 3 or 1 or 2 and you have a pool of more than 8, you will suffer from that. You have to follow exactly the barcoding schemes that the MGI field application support will tell you. And to be honest, at the beginning, I thought it's like Illumina does not really matter too much number of barcodes and multiplexing, but no, you have to have the sets of barcodes, the compatible barcodes all the time. And that is in the manual of the FS kit.
Those are all the questions. Oh, we have one more. Bala is saying, thank you to Dr. Ashraf for providing lots of insights on your experience with the V5.
Thank you.
Do you use dual barcodes?
Not at the moment, no. We don't use it because we didn't really see a need for it. We didn't see evidence of index hopping or barcode hopping, and we prefer the enzymatic method. So the option we have right now with the MGI is the classical single barcode approach. However, with different types of all these samples we've done now was blood samples, so the DNA quality is high. However, when we are moving to DBS or paraffin, it's better to use the mechanical shearing, and then we will be able to use the UDB and the dual barcodes.
Okay, we have one more question. This is from Dr. Akbar. Great presentation, Dr. Ashraf. Will you be publishing the case studies soon?
Well, it is really difficult because we are a commercial service. I cannot offer present case by case. because of patient privacy, etc. And it will be done through the collaborators and the clinicians who send us samples. They will drive that. So this is the difference between diagnostic and research lab. I don't really control the publication of the data.
Okay, does anyone have any more questions? What is the effective capture rate? What was the effective capture rate of MGI V5 probes with your library prep protocol?
Our average over the data I have so far, 150 exomes, was 71%. We reached 75, and down as the lowest was 65%. So 70% is what we expect with the capture, as long as you maintain accurate incubation, the accurate temperature at incubation times. It's very crucial. So the key here is to always calibrate your heat blocks and make sure that they actually display the recommended temperature. Otherwise, it will play havoc to your capture rate. So this is the key. We found that the key element in the capture efficiency, as well as the bite-in beads. We use the My1 M280, and we are shifting to the My1 streptavidin bite-in binding beads, and keeping them fresh. They go off pretty quickly, and their efficiency can change between runs. So this is another key factor in maintaining a high capture rate.
Okay, and then we have one more question. Do you have any experience with prenatal exomes?
Not with the V5, but it's doable. You can, as long as the prenatal sample is well withdrawn, and that is the reason I haven't reused it here, because it depends on the quality of the sample withdrawal by the clinician. And as anybody who worked with prenatal, you have to have your maternal contamination test, etc. So if you have majority of maternal blood in your sample, you'll end up with uninformative exome. The exome will work because you'll have the amount of DNA, but whether it represents the fetus, this is the key question. As long as you combine it with an exome of the mother, DNA of the mother, maybe a saliva sample or something, then you will be able to pick up the fetal variants. Okay.
Okay. Looks like there are no more questions. I'm just gonna check again. Okay. Yes. So, what other applications from MGI would you really want to try to use in the near future?
I think we tried everything. We would like to try the P300. I think the PE300 kit for the metagenomic. But for the human genome and the human exome, I think that the V5 is excellent. The library kit is reliable, and do not really, even the PCR, you do that, we don't get many duplications. As Akbar said, it's in the 8% region. It does not really offer a lot of duplication. It's just the sample quality. It's very sensitive to sample quality. So that's maybe the issue with the library kit. Other than that, the application I would like to try is the PE300, and maybe the cool hot, whatever they call, hot MPS or cool MPS, for the sequencing technology.
Okay, maybe we'll just wait another couple of seconds if anyone else has any more questions. I think that's it for now. Thank you so much, Dr. Ashraf, for your presentation and for your time. We really do appreciate you being online today with us and giving us an insight into your experience and being our customers.
Thank you, Rishma. Thank you for the opportunity to present our experience to your audience. And hopefully the presentation was useful for as many audience as possible.
Thank you. And before we end, I would like to thank the audience for joining us today as well and taking the time to listen to both our presentations. We appreciate your time and we look forward to welcoming you for our upcoming webinars. We have one coming up based on agriculture, which is talking about harnessing the power of genomics in agricultural, in the agricultural section, on the 20th of June 2023 at 10 a.m. Central European Time. And then we have the next one after that coming up on the 5th of July 2023 at 10 a.m. Central European Time, talking about oncology and the comprehensive tumor analysis by Chris, powered by MGI. And we look forward to welcoming you again for those two webinars on our Unlocked webinar series. So thank you again, everyone, for joining. We look forward to seeing you again soon.